Hello and welcome to the World Advanced Therapies and Regenerative Medicine Congress Interview Zone. My name is Freya Least, I'm the editor of RegMedNet and I'm your host for today. Now I'm very pleased to be joined by Mary Murphy, who's from the Remedy National University of Ireland Galway. So Mary, thank you very much for joining me today.
You're welcome.
So for those of you that may not know Remedy, can you tell us a little bit about it and also how you came to work there?
So Remedy is the Regenerative Medicine Institute and was set up by Frank Barry, Tim O'Brien and myself in 1995, which is quite a long time ago. Prior to that, both Frank and I had worked with Osiris Therapeutics in the US, where we gathered our experience in mesenchymal stem cells or mesenchymal stromal cells. And that was the basis, the cell basis of the Regenerative Medicine Institute. And in Remedy then we worked to produce the cells in a GMP compliant manner. We have a GMP facility. We work on the basic biology of the cells, and we also work to clinically assess those cells in early phase clinical trials. And that's a difficult thing to do. Producing cells is very, very expensive. At this point in time, we get EU funding for the many trials that we do for kidney disease, critical limb ischemia is another example. But in our specific area, osteoarthritis, we look to clinically assess using the cells and have been involved in a phase 1 clinical trial with European partners and currently doing a phase 2 trial following on from that, where we use autologous cells. And it's extraordinarily expensive.
I can imagine. So here at the World Advanced Therapies Congress, you're speaking on closed systems and automation. So what is a closed cell therapy manufacturing system?
Well, it's our solution to the expense of actually producing cells in an open, good manufacturing compliant facility, GMP facility, which is manned by a considerable number of people, very, very difficult work to do, and is costs a fortune. A single autologous batch could be up to €40,000 or maybe 35 sterling. So, and we were faced with that, doing our phase 2 clinical trial for osteoarthritis, it's autologous cells. And we looked at it and we said we have to go to automation. It has to be a way to take the cells away from human error and also the cost of retaining very many technical people in the facility. And that's where we started to think of automation and how we can do it. So,
interesting. So, I mean, when we have discussions around automation, closed systems are obviously always mentioned. But are there any other steps in the cell therapy manufacturing process where automation could be applied to save money and time?
So in our sort of thoughts of looking at automation, we did start at the collection of the sample. Yeah. And in the exemplar that we took, we were looking at bone marrow collection, which is done manually, and it's really quite a difficult thing and requires a lot of pulling to get the marrow out, and really requires a very experienced hematologist. So that's the first step we started looking at, and we sort of had a company come on board with us to actually design a controlled pressure system, which enabled the hematologist just to get the marrow harvest system, which is normally used anyway by hematologists, and have that be used in a controlled manner through an automated process and collected into a vial that's then ready with a tube set to input into a facility that will allow automated production. And that is our AutoStem system, which was developed over a 3-year EU-funded project again. And that's the system that we use now. I can describe it to you if you wish.
Yes, absolutely.
Okay, so what we've ended up with is it's almost a room within a room, glass-walled room for the most part. It has a base where we have supplies stored and some equipment sort of embedded in it, which has an automated port opening as needed. But again, once it's open, it's closed within the sort of the cell production areas. And in that area then we have three sub-areas. And in GMP production, you have to have different grades of sterility, and any open process needs a grade A system. Yep. So we have a grade A area for processing the produced cells. We then have closed processing developed with bioreactors. So we get our marrow, it comes in through the tubing system developed for the harvesting of the marrow. It's then directly inputted into a bioreactor, a closed vessel, basically glass vessel. That's all set up with our microparticles, and the cells then populate the microparticles. And with the right conditions, over 21 days, we can go from 30 mils of marrow to 1 billion cells.
Wow.
In a closed manner. That's our latest and greatest output. And that is being done in the Fraunhofer Institute in Aachen, which we built the system. So then that's the bioreactor, the grade D area, open, closed processing, so we don't need to have very expensive GMP environments and things like that. So, and once we have our microparticles loaded with the cells, we have to take them off, and we do that with an enzyme, and are then able to harvest the cells in various ways, really using a robotic-enabled and especially built tubing system again that allows us to transfer the solution of free cells into a closed area where we can then use a robot to transfer the cleaned up cells, essentially. We get rid of all the medium and can freeze them. And once they're frozen, they're transferred underneath the grade A area, excuse me, and stored in a freezer.
Yep. Wow.
And ready for use. So
is this a service which other institutions will be able to use, or will everyone have one of these AutoStem modules in their lab?
Eventually, I think we would envision it as coming to Galway, where we could test for clinical production and do some perhaps clinical trials. Yep. It could be purchased by anybody, I suspect, once it's all validated. And we can get a company to produce it, which should be fairly simple. It's not hard, all the elements are there. So we would hope that this would be available, 5-10 years for definite. Earlier, we hope, depending on funding. And we are currently looking for funding to go to the next step, to go from just MSCs, but also produce the vesicles that they generate. And to get the EVs, as we call them, produced in an automated manner. And that would allow much easier clinical translation. And as well as that, to go to the induced pluripotent stem cell derivation of the MSCs and the automated production of the MSCs, again to reduce the cost, along with automation to make sure that we have enough cells to treat patients when needed.
So that sounds very exciting. But for any researchers that maybe don't want to wait 10 or 15 years to have the wonders of automation in their lab, do you have any tips for them? What challenges might they face in trying to apply this to their processes?
Well, the challenges that any lab faces is scale, essentially. I'm describing the allogeneic scale. There are autologous systems that are being developed by, for instance, a company called Viva Biocell in Italy, and they're not fully automated, but they have a really nice plate-based system which will allow autologous production or even small-scale allogeneic production of the cells. So there are companies coming into the frame of automation for at least MSC production. But I think eventually going to a fully closed automated production unit will be the way. So there's no human error, we hope.
Fantastic. So Mary, obviously we hope we'll have the system in 10 to 15 years. What else can we expect from Remedy in the next few years?
Well, as I mentioned, hopefully going from just the production of bone marrow cells, I think it's easy to go to the production of cells from other tissues. That's not an issue. That would require perhaps, as you mentioned earlier, some development of automated systems. Now that they're available for adipose tissue, there's a company that does a lipojet, which seems to work fairly well. And I think really to try and miniaturize what we're doing in a way, optimize the production of the cells. And I think when companies come in and see that it is the future, things will happen much quicker, we hope. So.
Fantastic. Well, Mary, thank you so much for joining me today.
Okay, thank you.
And thank you all for watching. We'll be back with another interview from the World Advanced Therapies Interview Zone very shortly.